Today, we're going to hack the code of life. But here's the thing. This isn't a story about some futuristic, sterile lab. Nope. It's about an ancient, microscopic war that just might hold the key to our entire future. Let's dive in.
I mean, just think about it. This is one of the biggest questions in all of modern medicine. What if we could literally go into our own genetic code and fix the typos? You know, the tiny little errors that can lead to absolutely devastating diseases. I know it sounds like something straight out of science fiction, but the answer is surprisingly close to home. And here is the big twist. This revolutionary technology, it wasn't invented by humans in a lab. It was discovered. It was found hiding inside the tiny organisms that live all around us. And yep, even inside of us. So, to really understand how this all works, we first need to meet the incredible, revolutionary tool that bacteria have been using for, well, basically forever. It's called CRISPR. Now, that is a mouthful, I know. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. But the concept behind it is actually beautiful in its simplicity. See, long before we thought of it as a gene editing tool, CRISPR was and still is a natural immune system for bacteria. It's how they fight off viruses, and let me tell you, it is ruthlessly effective.
So, how did we even uncover this amazing tool? Well, our story doesn't start in some billion-dollar research facility. It starts with the clues that were hidden inside a single, very common bacterium.
And this, right here, is where the real detective work begins. Scientists had to zoom way in, focusing on one specific organism to try and decode its ancient secrets. So let's meet our main character, Lactobacillus pentosus KCA1. It's a pretty common bacteria, but this particular strain is fascinating. As the research paper points out, it was actually isolated from a healthy woman, and scientists took its entire genetic code, its whole genome, and put it under the microscope. So what did they find inside that genome? Well, get this. They found something incredible. The best way to think about it is like a genetic mugshot book, a catalog of every single enemy this bacterium has ever fought off. The very first clue? Scientists didn't just find one of these CRISPR systems, or arrays, tucked away in the DNA. They found 6 of them. 6! This wasn't just some simple lock on the door. It was a full-blown, high-tech security system. Okay, so take a look at this. This diagram from the actual paper shows us exactly how that genetic library is organized. Now, it looks a little complicated, but the whole structure is kind of like the spine of that mugshot book. And the individual pages, well, they're made up of 2 really key parts. First up, you have what scientists call direct repeats. In our little analogy, these are like the identical plastic sleeves, or the frames in the mugshot book. They create the structure and separate one bad guy from the next. And then, nestled right between those identical frames, you have the spacers. And folks, this is where the magic really happens. Each spacer is a unique snippet of DNA, a genetic memory that was literally stolen from a virus that tried to attack the bacterium in the past. It's a mugshot of a wanted criminal, written in DNA. So just how extensive is this criminal database? You're not going to believe this. The researchers identified a grand total of 118 unique spacers. 118. That's a historical record of 118 different attempted invasions that this bacterium survived and remembered. And here it is, the smoking gun. The researchers basically took these genetic mugshots and ran them through a database of known criminals. And as the paper says, they got hits. The spacer DNA perfectly matched the DNA of known viruses, also called bacteriophages, and other invaders. This was all the proof they needed. Okay, so having a book full of mugshots is great for identifying your enemies. But how does the bacterium actually fight back? Well, for that, it needs some muscle. It needs an enforcer. And that's where the Cas9 protein comes in. This is the molecular machine that does all the dirty work. It grabs a copy of a mugshot, that spacer DNA, and patrols the cell. If it finds any DNA that matches the mugshot, it acts like a perfect pair of molecular scissors and just snip. It destroys the invader on the spot. And here is what that enforcer actually looks like. Pretty incredible, right? This is the predicted 3D structure of the Cas9 protein from our specific bacterium. It's a complex, unbelievably powerful piece of natural machinery. Now, check this out, because this is why this particular Cas9 is so interesting. On the left, that's our guy, the KCA1 Cas9. On the right is a much more famous version of Cas9 that you might have heard of, See the difference? Our KCA1 version is just one single elegant chain. The other one is made of 4. It's way more complex.
So why does that matter? Why is simpler better? Well, if you're a scientist trying to use this as a gene editing tool, a simpler, smaller machine can be a whole lot easier to deliver into human cells. Discovering this new, streamlined version is a really, really big deal. So, okay, we've gone super deep into the genetics of one tiny little bacterium. It's time to zoom back out and ask the big question. What does all of this mean for us? What it means is we are moving from an era of just treating symptoms to an era of actually fixing the root cause. Where we used to see incurable genetic diseases, we now see the potential for precise, targeted gene editing. And the source of these new solutions isn't just human ingenuity. It's a massive toolbox we're borrowing directly from nature itself. And that's why this one bacterium, Lactobacillus pentosus KCA1, really matters. It expands our options. Every new CRISPR system we find is another potential tool for our toolbox. And sometimes a simpler tool, like this one, might be the perfect one for the job. It's just this powerful reminder that millions of years of evolution have created solutions we are only just beginning to understand. And that leaves us with a final, kind of mind-blowing thought. We are essentially taking the weapons from an ancient, invisible war between bacteria and viruses, and we're repurposing them to fight our own battles against human disease. The next great medical breakthrough might not come from a sterile lab, but from the secret, microscopic world that's been all around us this whole time.